Sandeep Chaube. Briefly, Sandeep did his PhD at Brandeis. He is basically a biophysicist by training. Subsequently, he did postdoctoral work in Harvard and then at MPI PKS in Dresden before joining IMSC roughly 2.5 years ago. So, Sandeep, looking forward to your talk.
So, thanks a lot, Arijit, for organizing this conference and as well as inviting me to give a talk. It's been really nice to listen to some of the things that we are kind of getting into, but of course, we do not have the expertise. We're just dabbling at some of the things. But I'm going to talk about something more biophysical. So the talk will be quite distinct from some of the talks that we have heard so far. Let's see if I can convey what we are doing and if it makes sense and so on. So it's an ongoing piece of work about how the genome is organized. So in general, of course, it's a bio audience. Of course, you are all aware of the fact that we have this. If we take the human cell, let's say it has a DNA which is about 2.2 meters long, and you compact it in a space of about in a kind of a spherical kind of cell of diameter 6 micrometers, right? And that is, of course, for people like us, it's kind of an interesting thing. How do you actually package them? And because what are the physical principles that govern this packaging? So a lot of work has been done, in particular people coming from a physics background or physical chemistry background have actually thought quite deeply about some of these problems. So I'll talk about a certain perspective that we are trying to build here. So this is, by the way, this is also true even in bacterial cells. Like you have these nucleoid-associated proteins. They can compact different parts of the DNA. So although here in eukaryotes, it's much more sophisticated, but you do see packaging here at the level of bacterial cells as well. Now, the perspective that I want to kind of bring in is this, like I would say about last 15 years, people have started observing that various proteins as well as enzymes in the cells form these kind of foci. And of course, the physical properties of these foci vary a lot. Often people call them liquid, these bodies as formed through liquid-liquid phase separation, but the phrase might not be quite correct. We are still trying to understand what the physical principles that govern the formation of these foci. Now, I will talk about two particular proteins. So in this experiments like MED1 and BRD4, these are proteins that are associated with transcriptional regulation. And as you can see, while they form these foci inside cells, if you take them out in vitro, you purify them, and you change the concentration, what you see is that beyond a certain concentration, they form these very spherical-looking droplets. And so they behave a lot like a mixture of oil and water. So if you take a bowl of water and put some mustard oil or any other oil, what you'll see is that there are these spherical structures that form. They may not form always a completely spherical structure, but something like this. So in physics, of course, it's been physical chemistry. People have studied these phenomena for more than 100 years and something called phase separation. But there are interesting differences in biological systems. One is, of course, that we are talking about a number of molecules which is equal to the Avogadro number. So it's really these are huge macroscopic structures. On the other hand, if you're thinking of proteins, typically you're thinking of hundreds to thousands to millions of molecules. So it's not entirely clear if you can use the same language as what we have used in traditional physics or physical chemistry literature and understand what's happening in biology. Now, one kind of potential mechanism is that these nucleoid-associated protein structures or these kind of different domains that you find in eukaryotes form through some sort of phase separation, right? It's a new kind of idea that is gaining a lot of ground these days, and we are trying to understand how that will be the theme of my talk. We'll try to kind of probe some of these aspects. Of course, probing them in vivo is challenging. So often what people do is that they will do these very simple in vitro studies. What you do here, you have a cover slip on which you tether the two ends of a DNA, and this is typically lambda DNA. That's what people use, and you flow in a certain protein, and you see that the protein and DNA together form some sort of structures, agglomerated structures, right? And this is what you see on the left-hand side. And similarly, you can look at an optical tweezer. You would trap any DNA between the two beads, and you again flow in proteins, and again you can see these structures. And these are done for two different proteins, which are FOXA. One is a binding factor, HP1 is again a heterochromatin protein, and so on. Now, one interesting thing is that when you form these structures, like these kind of protein DNA structures, they also apply forces. And of course, borrowing this term from the physics or physical chemistry literature, again, we can think of them as capillary forces, right? I'll get to some of the details. Now, the question that we like to think about is, of course, what we know that these experimental studies are saying that protein DNA together can form these structures, but DNA is, of course, heterogeneous. It can happen due to the actual sequence or at methylation patterns and so on. So we want to understand what the DNA sequence does here in this picture, right? That is the main question. For that, we construct a very simple coarse-grained model, and it's a control case. So what we have is a simple DNA polymer, which is again guided by the experiments, are tethered at the two ends, and this polymer is homogeneous for protein binding. In other words, these green molecules, which are proteins in the system, they bind to the DNA monomers with a certain affinity, and they can also interact amongst themselves with a certain affinity. So essentially, I have two variables in my system. One is a protein-protein interaction, and one is a protein-DNA interaction. But you'll see that leads to some really rather complex behavior, which we actually do not fully understand. But the first thing that I want to show is that if you simulate it using a Brownian dynamic simulation framework, is that the proteins like to kind of bind the DNA, and since they also like each other, they will form some sort of a condensate structure. But this is quite different from the water oil picture that I showed you, where it's a binary component, because you just have water molecules and oil molecules. But now you have a DNA as well, some polymer, which is again tethered. So that brings in its own complexity. Now, you see two things here. One is that they can start with multiple such condensates, but eventually they always lead to one. Now, this is actually, again, this also happens in oil-water mixture, and it's not known as Oswald ripening, something that happens that the system wants to minimize the total surface area of these condensates. So if you form these multiple condensates, they will all eventually merge and form a large condensate, and that is a known fact. And it turns out that happens even with the DNA, right? Moreover, you'll see that they form these condensates, but they also tend to move towards one of the ends. And this is something we see all the time. So that we think happens due to the fact that it gets more stabilized the more it goes towards one of the ends, right? Now, this is, for different concentrations, you can see what the profile looks like, and this is kind of what you see, right? It's just three different concentrations that we show. Now, let's actually think of a simple heterogeneous DNA model. And the model is the following. What you do is that you take the same average affinity as the homogeneous one, but you decrease the affinity of the flanking region, and you have a higher affinity region in the center. This is again some sort of a control case. We want to understand if affinity is higher in the middle, one would expect that a condensate should form in the middle, and that's actually what we see. That eventually you see that there can be multiple condensates, they come and merge and form a condensate in the middle. And so essentially the sequence or actual kind of heterogeneous pattern that you choose for your simulation seems to dictate what's happening. Of course, we know that DNA is not that simple. Of course, the heterogeneity is much more interesting for a DNA. But on the other hand, as you can imagine, that when this condensate is formed, it will apply forces to the flanking DNA, because it really wants to grab all the DNA it can inside the condensate, and that's why you see a force. That is really the source of the force. And the force is always felt at the interface of these condensates. Now, one thing that we can do is to actually change the end-to-end distance. Now, this is something experimentally you can actually very easily do. You can change the end-to-end distance and see how the forces will change. Now, for a homopolymer, the homogeneous DNA model that we had, you see a very slight increase in forces, and that has to do, we feel, is because of the size of the condensate and so on, because there is some change in the size of the condensate, but you see a minimal change, right? Really small. See about from 3.5 piconewton to some really 3.7 or 3.8. Now you see what happens with a heterogeneous DNA. What happens is that as you change the normalized end-to-end distance, like normalized end-to-end distance means like we take the end-to-end distance, we divide it by the contour length of the DNA. And then we, as a function of that, so of course, that does the normalization, the job of normalization for us. And as you increase the normalized end-to-end distance, after a while, it doesn't show much increase, but after a while, you see that there is a sudden jump. And this sudden jump can be understood from the fact that as you are changing the end-to-end distance, the DNA initially here, when the end-to-end distance is small, most of the yellow and red region, which are the high and low affinity regions, are inside the interface. And at the interface, you just have the yellow region, which is the low affinity region. Now, as you increase the end-to-end distances, suddenly the size of the condensate will be smaller, but you will have at the interface the red region. So because of that, there is a sudden jump because the red region has a higher affinity towards the protein. So the proteins really want to have more of the red region. So that's why this jump, right? So the forces do depend a lot on the actual interfacial sequence of the DNA. Now we can take a more complicated piece of DNA, and I'll just talk about one result here, which is going to be important for when we try to talk about some of the actual data. Here, what we do is that we have a rather kind of repeated structures of, this is often in the polymer physics literature, people will call them block copolymer. But what you have essentially is a high and low affinity regions interspersed in a periodic manner, right? And as you can see, I'm not going to show any simulations. The key result here is that, of course, you can do it for different concentrations, end-to-end distance, and so on. But what you see consistently for different conditions, under different conditions, not always, that you can actually sustain multiple condensates. Now, contrast it with the homogeneous DNA model that we talked about, where you can only see one condensate, no matter what the interaction strengths are, no matter what end-to-end distances are, it will always see one. But here you can actually sustain multiple condensates, right? And that has to do with the fact that these, again, if I'm thinking of just water liquid, this again, let's go back to that water liquid picture, then you would like to minimize your surface area. And so if you have smaller condensates, they will exchange molecules through diffusion. That's the only way they can talk to each other. But here, they're also talking through the DNA. So they're actually pulling each other. There's a tug of war going on between these two condensates. They're trying to pull DNA inside each other. And because of the tug of war, there is an additional way of interaction that emerges. And that leads to a rather complex behavior, which essentially can lead to sometimes the emergence of multiple condensates, right? Now we can actually take the lambda DNA, right? And then we call it a partial lambda DNA model. What we do is that lambda DNA is about 48 kb long, and that's what people do use in experiments. That's the motivation. And then we take 5 kilo base pair of a lambda DNA. This is how it looks like. As you can see, for the AT content or GC content, obviously, it will be the opposite. You will see it's very rugged. It's very noisy, rather than this blocky structure that we used in our simulations, right? Now, if you use this and use some sort of you say that the binding energies are proportional to the AT content. There are proteins that bind AT-rich region and GC-rich region. We're just taking one such example. And you see that the capillary forces, again, show a rather really non-monotonic behavior. In other words, you have if you change protein concentration, you see from this value of about 60 to about some 75 or something, you see a sudden jump, right? So initially when I saw the results, I thought that maybe something is wrong with the simulations, but actually it turns out that it's pretty reproducible, and we could actually get some understanding of it, which is the following. So if you look at the interfacial region of the DNA between these two, what happens, the only difference between the two is that as you change the concentration, the size of the condensate increases by some amount, but not by a lot. So the interfacial DNA, some monomers go inside when you go to this conformation this conformation to the top conformation. And because of that, the interfacial DNA that gets exposed this is kind of the interfacial part. And of course, it's not a single monomer because it's thermally fluctuating. So you'll get different monomers, you'll encounter different monomers at the interface at different times. And if you look at the average affinity, there is a difference of about 0.3 kbt between the two. And that is why you see this sudden jump, right? So what really matters is the average affinity. Now contrast it with a single molecule picture. If a single molecule binds to DNA, it will go and bind. That's a conventional picture, that it will go and bind to the strongest sequence. Here, because of the collective behavior of the proteins and DNA together, it seems that a lot of the physics is dictated by the average affinities, be it where they form or be it the capillary forces generated due to the interface. Now, let me actually show you that it's not all just some model churning, but there is some actual use to it. We looked at this particular paper. It just came out last month from our collaborator only. So what they have is this protein called DPS, which is an E. coli DPS. It's a nucleoid associated protein, and it's known to bridge different parts of DNA. So it binds different parts of DNA, but it's also kind of, people can kind of mutate and so on. So it's a protein that easily you can play with. So that's why it's used quite a lot. What we saw is the following, that again, they do the same experiment. They have a solution and DNA is tethered at the 2 points on a cover slip. You flow in proteins and you see that they form a single condensate, and that too at the end. And this happens consistently in all of the data. I'm not showing that data, but it's every image has the same thing, right? With or without flow, they'll always form at the end. And this is exactly akin to what we see for our homopolymer simulations, right? Turns out that indeed DPS is a protein that bridges DNA, that binds DNA in a sequence-independent manner. It doesn't have any sequence dependence, and which is consistent with what we see in our simulations. Now let's look at another protein, of course, SOX2. Now SOX, of course, is a pioneer factor protein, so it's one of the Yamanaka factors. Now, if you look at SOX2, it does form multiple condensates here. And this is another experiment. It's again a recent experiment 2 years back, 2 years before it came out. And you see there are multiple condensates. And remember what I told you about heterogeneous DNA. You cannot sustain multiple condensates without having strong sequence specificity, right? So it's so what it tells you is that really it's a sequence-dependent co-condensation that we are seeing. Now, let's look at another protein. It's called HP1. People typically think that HP1 is a heterochromatin protein. It bridges DNA again. People typically think that it binds in a sequence-independent manner. That's kind of the usual understanding. But what we see is that if you look at them here, you will actually see that there are two condensates. And if you have two condensates, it cannot be explained by a homogeneous model. So clearly there is some level of sequence dependence that it has. So many experiments looked at many of the experiments that they do not see a lot of dependence, but whatever the little dependence that it has to the DNA, sequence dependence it has, that clearly plays a role in the co-condensation of these proteins. Okay, so I'll briefly talk about, again, an ongoing collaboration with Mahipal Ganji's lab from IISc. And Mahi has been, and his students have been looking at this particular protein, that LSR2, which is a nucleoid-associated protein in bacteria. And what you see is that if you do an AFM image, if you look at these images, what you find is that in presence of DNA, they are forming these really bright foci. And yellow is what you tag these are like essentially the proteins. These proteins essentially take different parts of the DNA and bridge them, right? It's like really forms this kind of concentrated structures. Now we want to understand how this is happening. What are the physical principles? So for that, and in particular, we also want to understand the role of DNA sequence. And the first experiment they did is that they changed the concentration of proteins. So just essentially in a test tube, you put a lot of proteins and you keep changing the concentration and you see what happens, right? And pardon me the color, these experiments were done just a couple of weeks back. And so it's the same thing. Just the colors are different. Don't get distracted by that. So what you do see is that if you change your concentration from 100 nanomolars to about 10, like 1 micromolar, you start seeing these droplets. And from 10 micromolar onwards, you see these really large droplets. Okay, so I'll be done before that, so it's fine. So these proteins, what it suggests is that these proteins actually like each other, so they attract each other and they form these kind of structures. But interestingly, if you add DNA, some 0.6 nanomolar DNA inside this, you start seeing structures even at as low as 100 nanomolar concentration. Now, it's very similar to the picture of this protein-mediated condensation of DNA, because they are also, if you add proteins, you can actually condense DNA at a much lower concentration. So that was kind of interesting. So what we thought is that let's actually to the usual magic he does, which is again that he kind of tethered the two DNA and the DNA at the two ends using this on a cover C based assay. Okay, and what they see is the following. So if you just look at the DNA, this is just the DNA. So the DNA actually makes this compacted kind of regions, as you can see. A lot of them tend to form at the end, and that we think has to do with the fact that they are tethered at the end. So there is always a competition between your energy and like binding energy and this advantage that you get, the stabilizing effect that you get at the tethered ends. On the other hand, if you look at the actual protein, they seem to co-localize. Of course, this is one rough image. I didn't just wanted to give you a flavor, but you can tag both simultaneously and see what's happening. Right? Now, again, I'm not going to talk about it in any detail. Just wanted to give you a flavor. So they constructed a piece of DNA with this kind of 80 content being the pattern. So you see here the x-axis is the DNA length. This goes up to 17.5 kb, and then this is the 80 content as you go along from 0 to this value, right? Now, we set up a simulation which mimics this sequence, and this is what we see. And in experiments, this is the kind of pattern we see, right? So essentially, what it establishes is that you have like the actual sequence. It's not just co-condensation, but it's actually the sequence that dictates the position of the co-condensate. I mean, there is a lot to it. I'm not going to tell anything more than this. So I'll just conclude by saying that homogeneous DNA will always lead to a single co-condensate at one of the tethered ends. And then heterogeneous DNA can sustain multiple condensates and can lead to some sort of a pearl necklace-like structure where you have multiple condensates along the DNA. Then it's the average interfacial DNA affinity that dictates the capillary forces, which has implications for genome organization, because often people think of forces being required for packaging a certain genome, and if you can change forces by such large amounts, by just changing the concentration, what it really implies is that rather than changing the whole DNA, even very local changes at the motif level. So if you have some mutations and so on, or some methylation related changes, if you change the affinity of the interfacial DNA of even about 10 to 50 base pairs, you can change global like forces at the global level at the kilobase pair level. So that is one. And then we find that LSR2, which is one of the nucleotide associated proteins, seems to package DNA through co-condensation, the kind of mechanism we talked about, in a sequence-dependent manner. So that's it. I would like to thank Rohit and Pinaki. Rohit is a PhD student. Pinaki is a postdoc. They did it together, but a lot of the work was done by Rohit. And also our computing facility. And then Mahipal, Tejas, and Prakshi for assisting us with the experiments and also a lot of experimental input. Thanks a lot.
Thanks, Dave, for this wonderful talk. So we have time for questions.
Yeah, that was very interesting. Just if you can go to your conclusion slide.
Yes.
This is something that's puzzling me.
Yes.
And this is, I'm looking at the central picture in which there are two blobs on either side.
Yes.
But there is also a red strand in between.
Yeah.
This is a picture that you had also shown earlier.
Yes. So we are just coloring the strands with their affinity.
Yes. So why is that? Why does that strand not have a blob on it?
Exactly. So that's a very good question we do not fully understand. Now, of course, that's a high affinity region, but it's really, you see, the physics is really complicated, because what complicates it is that if you just untether the DNA, then we really understand it quite well, because there is another project where we're thinking about just untethered DNA and then how the affinity affects things. There you understand it quite well. The problem is that at the end, these condensates are much more stable. Now, let's say there is a...
So why is that? That's
yes.
also not very clear.
Yeah. We are kind of looking at that. Again, it's not super clear to us, but it seems that it has to do with the time scales. Like if you are at the end, because you are tethering them, and it's a semi-flexible polymer, I didn't talk about the details. So what happens is that the mean square displacement is much less. So if you have proteins that bind, effective off rate is less at the end compared to the middle. So essentially they work as absorbing boundaries. So that's what we think is happening. We do not know. We have to kind of explore that a bit more. that effect combined with the affinity effect makes it rather challenging to see exactly where they're forming. So and again, problem is that if you do not have an analytical theory for it, one needs to write down a proper field theory for this. Otherwise, the difficulty is that if you're just running many how many can you run? That is one of the limitations. But we do not fully understand that. But it seems that if you see, both of the condensates do contain the red region, high affinity region, but there is one inside that doesn't seem to. And it's a repeated pattern. You will always see that if they find these high affinity regions at the end, they tend to form at the end. But if something is in the middle, and this is something we also see in the data, by the way, like if you look at LSR2 data and so on, you'll see that there are multiple condensates at the form, but they always tend to form at the end. So, but there won't be a single, there will be multiple, but it's always preferred to bind at the end as well as high affinity region. So it's an interplay of the two. That is what dictates it.
Again, I have many questions, maybe detailed, I'll talk to you later.
Yeah.
But two things. One is, what happens if we change the protein as a homogeneous protein you got here? So if you make it, the protein is a homogeneous one, the green thing, but it's the charged parts of the protein that usually first interacts with the DNA in terms of the phosphates. So if you change your protein, instead of making it a homogeneous one, but have some heterogeneous surface, maybe some charged regions and non-charged, does it affect the whole?
We haven't done that. Again, I would say the same thing, that all these effects are there, right? I mean, we do not deny, and you are absolutely right, that it also might be that not every, you can think of the proteins as patches. Some patches interact more strongly than some of the other patches, but we do not consider that. Because we are having a hard time even to understand this model properly.
Yes, so, and have you tried a circular DNA?
No, not yet.
Yeah, right. Very nice, interesting, thought-provoking, I would say. But yeah, so what about the stoichiometry of these? Is it comparable to let's say, the proportion of protein and DNA that are found in, let's say, some of the condensates?
Yeah, that's a very good question. Yes, so we choose the parameters like all these micromolar. So these are parameters that are chosen from these HP1 experiments. And so some of the experiments people do in actual conditions are those are the ones that motivated us to choose these parameters. So they're comparable. That's why we can talk about piconewtons and so on. Otherwise, it kind of doesn't quite make sense.
And the size of these beads, so right now, you did show a plot where I think it was given. So each bead is, what is the...
Yeah, so the way we think of it is that, so essentially we took 5,000 base pair long lambda DNA, and we say that if we take many of the proteins tend to occupy some, of course it varies, but let's say as a footprint of about 10 base pairs, roughly, then you'll have 500 monomers, which is kind of also for us in simulations easy to do. That is also another key factor. Now, so essentially, so each protein has a size. So just think of that. So each protein occupies one monomer, and that one monomer has 10 base pair footprint. That's how we see it.
Okay, so each it has 10
exactly.
10 base pairs. Okay. So I understand that this. Okay.
Go ahead.
So I understand that this is the very clean experiments in terms of tethered DNA, but in real systems, what would be an approximation? So in what approximation would this fit in is what I'm trying to understand.
See, in real systems, in general, if you ask me, I would say that, this whole field of LLPS has that criticism, that how do you test these ideas in vivo, right? And that I think is a very valid criticism. At this point, I do not have a very clear answer to this question. But I think, the first thing, so we have been kind of trying to do some of that, but one needs to first understand what's happening with these minimal systems, I would say, and then try to look for certain signatures that we find here to see if you can see them there as well. Like, for instance, so like one of the work which is kind of related to it, but not exactly, there is something called pre-wetting and some sort of wetting transition that we saw. So that was my postdoc work, and there we looked at KLF4, which is one of the Yamanaka factors again, and it seemed to kind of actually form some wetting layer on DNA. And that was something that we, again, studied. We had an in vitro setup, we studied it and actually built some model and so on. But then now there is, so some of these people actually ended up testing in vivo. So they looked at some data and then trying to find some of the signatures that we found in vitro setups. But first, I think it's very important to establish the physics of it, and then you kind of go on and do that. But still, you're far from this. That's what I'll say. You're not yet there.
So you said it binds more preferentially to AT-rich regions. So what is different about AT-rich regions? Because the diameter of the DNA is the same. And the physical properties of the DNA would be the same between AT-rich and GC-rich regions. And is there any denaturing of the DNA? Because there's a difference between AT and GC-rich DNA.
Yeah, so again, I think it goes back to some of the things that SK was saying, that I think, of course, so then there you have to look at the actual structure, and also I think Ashutosh was talking about. So you have to see like how the protein binds, what are the DNA binding domains, and how that happens. But of course, in this model, we are agnostic to that. Actual molecular mechanism, we are agnostic to that. We just say, let's say they bind, and then we kind of see the effects of the consequences. But you're right, I mean, we do not know that. And of course, the other thing is, I think as you was kind of pointing out, so there is this really nice study that also changes that sequence also has an effect on bendability. So you can write a code for the DNA without a sequence, but just by looking at the bendability of the DNA, just the polymeric mechanical properties of the DNA. And that I think was, so you can use this in a parallel manner, in an orthogonal way. That I think is also quite interesting. Now, whether those things will have a role, I'm sure that they have a role. I do not know. That's kind of.
Let's thank Sandeep for this wonderful talk.